Welcome to this video on Erysipelothrix, a genus of gram-positive bacteria known for causing erysipelas in pigs, turkeys, and other animals, and occasionally infections in humans. In this session, we will explore its key characteristics, growth patterns, laboratory identification, and clinical significance. By the end of this video, you'll have a clear understanding of how to recognize and study this important pathogen in veterinary microbiology. Genus Erysipelothrix. These bacteria belong to the group of regular, non-spore-forming gram-positive rods, along with lactobacillus and listeria. They are mesophilic, facultative anaerobes and are non-acid fast. Morphologically, they appear as short rods or long, non-branching filaments and may occur singly or in short chains. Erysipelothrix species are non-motile and negative for catalase, oxidase, indole, methyl red, and Voges Proskauer tests. They do not hydrolyze esculin or urea. They ferment glucose weakly without gas. A key identification feature is their ability to produce hydrogen sulfide in triple sugar iron agar. The genus contains two major species, Erysipelothrix ruziopathiae, which is clinically important and zoonotic, and Erysipelothrix tonsillarum, which is usually non-pathogenic in pigs but has been linked to endocarditis in dogs. Regarding habitat, Erysipelothrix ruziopathiae is widely present in nature. It survives well in low temperatures, alkaline environments, and organic matter. It can be isolated from many animal species, but pigs serve as the primary reservoir. Healthy pigs may carry the bacteria in their tonsils and intestinal lymphoid tissues. Both sick and healthy animals can shed the organism through feces and secretions, creating a major source of environmental contamination. This makes control and hygiene very important in preventing the spread of infection. Erysipelothrix ruziopathiae causes disease in many animals and is an important zoonotic pathogen. Infection usually occurs by ingestion of contaminated food or water, but the organism can also enter through skin wounds. This makes it an occupational hazard for veterinarians, butchers, and fish handlers. In humans, the disease is known as erysipeloid. It produces a localized, painful, violaceous cellulitis around the site of inoculation, often accompanied by induration and edema. Systemic spread and endocarditis are rare, occurring mainly in immunocompromised individuals. The organism is also one of the most common pathogens acquired from handling fish. Erysipelothrix ruziopathiae possesses several key virulence factors that help it survive and cause disease. The capsule, or glycocalyx, protects the bacteria from opsinin-mediated phagocytosis and intracellular killing. The SPA protein is an important surface antigen that contributes to protective immunity. The large surface proteins RSPA and RSPB support biofilm formation and help the organism adhere to surfaces like fibronectin and collagen. Neuraminidase removes sialic acid residues from host cells, playing a crucial role in attachment and the development of vascular lesions. Hyaluronidase acts as a spreading factor by breaking down hyaluronic acid in connective tissues, allowing deeper tissue invasion. Finally, superoxide dismutase neutralizes oxidative metabolites released by phagocytes, helping the bacteria survive inside host cells. The acute form of swine erysipelas presents as a rapid septicemia. Pigs show high fever, depression, loss of appetite, and the classic diamond-shaped skin lesions. These red, raised plaques result from vascular damage and thrombosis. Mortality is high due to widespread vascular injury and shock. The subacute form is milder than the acute stage. Pigs may show fever and reduced appetite, with fewer or less prominent skin lesions. Septicemia is present but less severe, and most animals recover with timely treatment. The chronic form develops after the acute or subacute stage. It is characterized by endocarditis and polyarthritis. Valvular damage leads to heart murmurs, while chronic synovitis causes joint swelling, fibrosis, and erosion of articular cartilage. Affected pigs show lameness and reduced performance. In turkeys, the infection is often acute and highly fatal, whereas chickens are only rarely affected. In turkeys, geese, and other birds, the infection usually causes acute septicemia, often leading to sudden death. Chronic cases may present as endocarditis or arthritis. In young sheep, the organism can enter through the umbilicus or wounds, leading to chronic polyarthritis. Adult sheep may develop post-dipping lameness, characterized by cellulitis that extends into the laminae of the feet. Occasional infections of varying severity are reported in dolphins, cattle, dogs, horses, lambs, and rabbits. Overall, the pathogenesis of Erysipelothrix Morphology revolves around vascular injury, immune-mediated tissue damage, and evasion of host defenses, driven by its diverse virulence mechanisms. For laboratory diagnosis, samples are collected from the liver, spleen, kidney, heart, and synovial tissues at necropsy. Recovery from skin lesions or chronic cases is difficult. In humans, a biopsy from the deeper layers at the lesion edge is recommended for best results. Microscopy shows different morphologies depending on the disease form. In acute cases, the organism appears as slender gram-positive rods, singly or in short chains. In chronic cases, long, unbranching filaments are seen, which may partially decolorize. Erysipelothrix Morphology is isolated primarily on blood agar, with selective media containing sodium azide and crystal violet. Plates are incubated aerobically at 35 degrees Celsius for 24 to 48 hours, with carbon dioxide enhancing growth. The organism grows between 5 and 42 degrees Celsius, tolerates alkaline pH, and survives in high salt concentrations, but does not grow on McConkey agar. Colonies of Erysipelas are small, non-hemolytic after 24 hours, sometimes showing alpha hemolysis at 48 hours. Smooth S-form colonies are convex and circular, while rough R-form colonies are flat and irregular. Microscopically, acute cases and S-form colonies show short gram-positive rods, whereas chronic cases and R-forms show long filaments that may appear partially gram-negative. Erysipelas is a facultative anaerobe with growth enhanced by carbon dioxide. It is catalase and oxidase negative, non-motile, non-spore-forming, and non-acid fast. It does not hydrolyze esculin or produce urease. On TSI agar, it produces hydrogen sulfide along the stab line. Fermentation is variable. Lactose, glucose, lebulose, and dextrin are usually positive. R-form colonies show a distinctive bottle brush growth in gelatin, though this is slow. Automated systems like API Corin and Vitect can be used for identification. Antimicrobial susceptibility of Erysipelothrix Morphology is usually determined by agar dilution following CLSI guidelines. Modifications, such as adding 5% horse blood, can improve growth. Specific breakpoints exist for drugs like tiamulin used in swine therapy. Erysipelothrix Morphology is treated mainly with penicillin for acute cases, while tetracyclines, lincomycin, and tylosin may be effective. Many antibiotics, including aminoglycosides and sulfonamides, are ineffective, and chronic erysipelas has no reliable treatment. The bacterium survives for months in the environment but is killed by heat, irradiation, and disinfectants such as quaternary ammonium and phenolics. Strain typing can be done by serotyping, but molecular methods like PFGE, ribotyping, MLE, and SPA sequencing provide better discrimination. PCR allows rapid detection from tissue or meat, identification of resistance genes like TET, M, and differentiation between species.